Hello students, this is Niharika, Assistant Professor, Department of Biotechnology from Government College Hisar, under the aegis of Director General Higher Education Haryana. And I'm going to discuss with you the topic antigens from the subject immunology. Before we start, let us take a look at what we are going to learn today. The objectives of this lecture are to understand and differentiate between antigen and immunogen, to learn what are epitopes, their significance and types, to discuss the factors influencing immunogenicity, to learn about the different types of antigens and their classification, to know what are superantigens, and to discuss briefly about adjuvants. Let us start with the introduction. So, what is an antigen? It can be defined as any substance that can bind specifically to the components or products of immune response, that is, antibodies or T cell receptors. Another term related to antigen and used interchangeably with the antigen is immunogen. It can be defined as any kind of stimulus that produces a humoral or a cell-mediated immune response in an organism's body. Antigenicity is the ability of an antigen to combine specifically with the products or components of the immune responses, and immunogenicity is the ability to induce or stimulate a humoral or cell-mediated immune response. Now you see, there's a difference between antigen and immunogen. An antigen can combine specifically with its corresponding antibodies or T cell receptors, but may not be able to produce or evoke an immune response, while an immunogen induces a humoral or cell-mediated immune response and also has got the property of antigenicity. Therefore, it can be said that all immunogens are antigens, but not all antigens are immunogens. An example of this is hapten. Haptens are small-sized or low molecular weight molecules that can bind to their specific antibodies or T cell receptors, but they are unable to initiate an immune response by themselves. So basically, haptens are antigenic, but they lack immunogenicity. But they can be made immunogenic by combining them with large carrier molecules. This will be discussed in more detail in later slides. How the antigen interacts with the components of the immune system. This is through epitopes. Epitopes are the small areas or the portions of an antigen that are recognized and bound by the specific antibodies or T cell receptors or MHC molecules. You can say these are the smallest units of antigens that are capable of binding specifically with the antibodies or T cell receptors. That is why they are also known as antigenic determinants, because they determine the antigenicity of an antigen. Now, corresponding to the epitopes are some sites in the variable regions of antibodies or T cell receptors that recognize, interact, and bind to the epitopes. These sites are known as paratopes. An antigen can interact with how many antibodies depends on the number of epitopes on the surface of antigen, which is called its valence. An antigen having multiple epitopes can combine with multiple antibodies at one time. An antigen having a single epitope is called as monovalent, while the antigen having multiple epitopes is called polyvalent. On the basis of their structure and stereochemical interaction with the paratope, the epitopes of protein antigens may be conformational or linear. Conformational and linear epitopes interact with the paratope based on the 3D conformation adopted by the epitope, which is determined by the surface features of the involved epitope residues. In this picture, you can see an antigen having three different types of epitopes. That is why this is a polyvalent antigen. These three different types of epitopes make it possible for the antigen to bind three different types of antibodies at a time. As I mentioned, the epitopes of protein antigens may be linear or conformational. A linear epitope is formed by the 3D interaction of sequential or contiguous amino acid residues, that is, the amino acids that are lying next to each other in sequence. However, a conformational epitope is formed by the 3D interaction of non-sequential or discontiguous amino acids, that are the amino acids that are far apart in the terms of primary amino acid sequence but come close together in the tertiary structure of the protein by protein folding. So the native protein conformation is very important for these conformational epitopes, because if the tertiary structure of the protein is somehow lost or degraded, the topographical structure of these conformational epitopes will also be disrupted. And most of the antibodies bind to the protein antigens in their native conformation due to these conformational epitopes. Take a look at this picture. Here in the figure A, you can see a linear epitope is bound to an antibody. In this linear epitope, there are continuous amino acids lying next to each other. In the figure B, there is a conformational epitope bound to an antibody. If you see the primary sequence of this conformational epitope, you can see that the amino acids that are making up this epitope are not continuous and there are distances in between them. So, by this picture, I think the difference between the linear and conformational epitope must be very clear. There are differences in the antigenic interactions of T cells and B cells. And based on this, there are B cell epitopes and T cell epitopes. In case of interaction of antigens with B cells, binary complex of membrane immunoglobulin and antigen is formed, while in case of T cells, ternary complex of T cell receptor, antigen and MHC molecule is formed. B cells mostly recognize and bind soluble antigens, while T cells mostly bind large insoluble antigens. In case of B cells, MHC molecules are not required for antigenic interaction, while in case of T cells, MHC molecules are an absolute requirement to display the processed antigens on their surface. B cells mostly recognize and bind protein, polysaccharide and lipid antigens, while T cells mostly interact with protein antigens. However, some lipids and glycolipids that are presented on the surface of MHC molecules may also get interacted with T cells. In case of epitope properties, the epitopes of B cells are accessible hydrophilic mobile peptides containing sequential or non-sequential amino acids, while in case of T cells, the epitopes consist of internal linear peptides produced by the processing of antigens and bound to the MHC molecules. Now, let us take a look at the different factors that determine the immunogenicity of an antigen. Or you can say these are the basic requirements for any agent to be immunogen. These are degree of foreignness, molecular size, chemical structure and heterogeneity, and ability to be processed and presented by an antigen presenting cell. I'll be discussing each one of these in detail in the upcoming slides. Now let us understand what is meant by the degree of foreignness. The immune system of an individual has got the remarkable property of distinguishing self molecules from non-self molecules. That means the immune system is able to recognize the molecules that are not a part of the body as foreign and they are targeted for elimination, while the molecules that make up a normal part of the body or belong to the body are not recognized and targeted by the immune system. They are recognized as self. So, in order to elicit an immune response, any molecule must be considered as foreign by the immune system. The property of being tolerant to the self molecules is acquired during the development of lymphocytes. So, greater is the phylogenetic distance between the organisms, greater are the genetic variations, greater are the structural differences, and hence greater is the foreignness of an agent. So, greater will be its immunogenicity. The same molecules that are recognized as self in an individual can act as immunogens in other individuals of same species because there they are recognized as foreign or non-self. The second requirement for being immunogenic is that the molecule must have a certain minimal molecular weight. There is a direct relationship between the size of the immunogen and its immunogenicity. Most of the good immunogens have a molecular weight of 1 lakh daltons or more. In general, the small molecular weight agents or molecules having a molecular weight of less than 1000 daltons are not immunogenic, while those having a molecular weight between 5 to 10,000 daltons may or may not be immunogenic or are poor immunogens. In short, relatively small substances have decreased immunogenicity, whereas large, high molecular weight substances have increased immunogenicity. In this table, you can see the molecular weights of some common experimental antigens used in immunology. KLH is having a molecular weight of greater than 2 lakh daltons. BGG has a molecular weight of 1 lakh 50,000 daltons. And the common BSA protein has got a molecular weight of 69,000 daltons. And then there are many others having different molecular weights. Continuing to the third factor, chemical structure and heterogeneity. Size and foreignness are not by themselves sufficient to make a molecule immunogenic. Another property required for immunogenicity is the presence of a certain degree of chemical and structural complexity. Very simple molecules such as homopolymers of amino acids and sugars are not good immunogens regardless of their large size. Among all the biomolecules, proteins have very high structural variations, high structural complexity, and different levels of organization, that is, 4-level organization structure: primary, secondary, tertiary, and quaternary. These contribute greatly to the immunogenicity of proteins, and proteins are among the best immunogens. Greater is the degree of complexity of the protein, greater is the heterogeneity, that is, the variation in the different types of molecules in the structure of proteins, greater is the immune response to that protein. Lipids and nucleic acids are generally poor immunogens. However, they can be made immunogenic when they are conjugated to proteins. Such lipoprotein and nucleoprotein complexes make up good immunogens. So, by this you can understand the importance of chemical structure and chemical variations or heterogeneity to the immunogenicity. This picture is showing the increase in the degree of immunogenicity with the increasing chemical and structural complexity of different biomolecules. And the maximum, as you can see, is for proteins. Last but not the least factor of immunogenicity is the ability of antigen to be processed and presented. Production of both humoral and cell-mediated immune responses requires and involves the interaction of T cells with the antigen that has been processed and presented in association with MHC class I or class II molecules. The macromolecules that cannot be degraded and presented with MHC molecules are generally poor immunogens. This is exemplified by the polymers of D amino acids, which are the stereoisomers of the naturally occurring L amino acids. Because the degradative enzymes within antigen-presenting cells can only degrade proteins containing L amino acids, these polymers of D amino acids cannot be processed and are resistant to enzyme degradation. So, they are poor immunogens. Large insoluble macromolecules are generally more immunogenic than the small and soluble ones because they are more readily phagocytosed and easily processed. So, you can see how the susceptibility of antigen processing and presentation also affects the degree of immunogenicity. Apart from these basic requirements of immunogenicity, the ability of an antigen to induce an immune response will also depend on certain properties of the biological system that the antigen encounters. Most important of these are the genotype of recipient and the dosage and route of antigen administration. If we talk about the genotype of recipient, it is the genetic makeup of an individual that influences the type and degree of immune response. The genetic control of immune responsiveness is largely determined by the MHC genes and their products, which function to present the processed antigen to T cells. Also, the response of an individual to an antigen is influenced by the genes encoding B cells and T cell receptors and by the genes encoding various other proteins involved in the immune regulatory mechanisms. The genetic variability in all these genes affects the immunogenicity of a molecule acting as antigen. Another important contributor is the dosage and route of antigen administration. An insufficient dose of antigen will not stimulate an immune response either because it fails to activate enough lymphocytes or because it induces a state of non-responsiveness called tolerance to the antigen. Also, the excessively high dose of antigen cannot induce an immune response because it causes lymphocytes to enter the state of tolerance. A single dose of most immunogens will not induce a strong response. Rather, repeated administration of doses of antigen called the booster shots are required for a strong immune response because they increase the clonal proliferation of antigen-specific T cells or B cells. Also, the route of antigen administration is important because it determines which immune organs and cell populations will be involved in the response. Differences in the lymphoid cells populating these organs generate differences in the quality of the subsequent immune responses. Now moving on to the types of antigens. Antigens may be categorized according to different criteria. First, we'll talk about the classification of antigens on the basis of origin. On the basis of origin, antigens can be exogenous antigens or endogenous antigens. Exogenous antigens are the foreign particles or antigens that originate outside the organism and enter the body from outside. These types of antigens are internalized and degraded within the body and subsequently presented with MHC class II molecules. Examples include bacteria, fungi, protozoa, free viruses, etc. Endogenous antigens are those antigens that originate within the organism. They may be antigenic products, subfragments, or compounds generated within the body. These types of antigens are generally proteins that are degraded into peptides and presented with class 1 MHC molecules. Examples include products of intracellular viral and bacterial replication and self-peptides originated from cellular proteins. Apart from the classes exogenous and endogenous, depending on their origin, antigens may fall into other categories also. Autoantigens. These are an organism's own or self-antigens that stimulate an autoimmune response in the body against themselves, resulting in autoimmune disease. These antigens under normal conditions should not be the target of immune system, but due to some genetic or environmental factors, the normal immunological tolerance for such antigens has been lost. The examples include self-proteins, nucleoproteins, nucleic acids, corneal lens protein, etc. Neoantigens. These are the antigens presented by MHC molecules on the surface of tumor cells. These antigens result from a tumor-specific mutation during transformation of normal cells into cancer cells. Alloantigen. It is the antigen found in different members of the same species. It is present in only some individuals of a species so that it stimulates immune response in those individuals that lack it. Examples include blood group antigens A and B and histocompatibility antigens present in the grafted tissues. Heteroantigens. Heteroantigens are present in different species. These are the antigens derived from one species that are able to stimulate immune response in other species. What are heterophile antigens? These are the identical antigens found in the cells of different species. These antigens are identical or similar in nature and structure that are present in the different and unrelated species, and being identical or similar, they are responsible for cross cross reactivity of antigens. Examples include Forceman antigen and cross reacting microbial antigens. On the basis of chemical nature, antigens may be classified as proteins, polysaccharides, nucleic acids, or lipids. Most of the antigens are proteins. They may be pure proteins, glycoproteins, or lipoproteins. If they are polysaccharides, they may be pure polysaccharides or lipopolysaccharides. Nucleic acids are generally poorly immunogenic, but they may become more immunogenic when they are in single-stranded form or complexed with proteins to form nucleoproteins. Lipids are usually not immunogenic, although they may act as haptens, and when conjugated with large carrier proteins, they may become immunogenic. How the antigens induce antibodies formation against them forms the basis for the classification as T-independent or T-dependent antigens. T-independent antigens are those that are able to stimulate the B cells directly for the formation of antibodies against them without the requirement of help from T cells or the interaction with T cells. These are large, complex, multivalent antigens with multiple repeating units, so they are able to cross-link multiple antibodies at their surface at a time and thus directly activate B cells for antibody formation. The immune response to these antigens is fast but short-lasting, as no immunological memory against antigens is formed, and the predominant class of antibodies formed is IgM. Examples include polysaccharides, lipopolysaccharides, and nucleic acids. T-dependent antigens are those that require the help of T cells for antibodies formation. Antigen-presenting cells recognize these types of antigens and present them to T helper cells, resulting in the release of cytokines, which then activate the B cells to produce antibodies against them. The response to these antigens is slow but long-lasting because antigen-specific immunological memory is formed, and the immunoglobulin classes formed are all types IgG, IgM, IgA, IgE. The examples include proteins. On the basis of immunogenicity, antigens can be complete or incomplete. Complete antigen is the one that is self-sufficient to induce a full-fledged immune response, resulting in the production of antibodies that will specifically interact with the antigen. These are generally high molecular weight antigens having molecular weight more than 10,000 daltons. They may be proteins or polysaccharides. Incomplete antigen or hapten. As I discussed earlier also, haptens or incomplete antigens are unable to initiate or induce an immune response on their own because of their small size or low molecular weight. However, to make them immunogenic, they need to be attached or combined with a large non-antigenic carrier protein molecule such as albumin or globulin, and after their conjugation with the carrier molecule, they are able to induce an immune response and antibodies are formed against this hapten carrier conjugate. Hapten then reacts with its corresponding antibodies. So the examples of incomplete antigen or hapten include capsular polysaccharide of pneumococcus and DNP, that is dinitrophenol. This picture is showing the conjugation of hapten molecules with a large carrier molecule, and this hapten carrier conjugate then becomes a complete antigen. There is another class of antigens called superantigens. These are very potent immunostimulators that can cause excessive activation of the immune system much more than the normal antigens. They interact with very large numbers of T cells and result in the polyclonal activation of a very large fraction of T cells, which results in the secretion of excessive amounts of cytokines. The superantigens bind directly to the MHC molecules without the need for antigen processing and presentation. Examples of superantigens include several types of bacterial toxins such as staphylococcal enterotoxins and streptococcal pyrogenic exotoxins. If you observe this picture carefully, you can see the difference between the immune responses to a conventional antigen and a superantigen. In case of a conventional antigen, only 1 out of 10,000 to 1 out of 1 lakh of T cells are activated, and the response is monoclonal or oligoclonal. But in case of superantigens, as high as 1 out of 4 to 1 out of 10 of T cells are activated non-specifically, and the response is polyclonal. So this is the power of superantigens. Moving on to the next topic, adjuvants. So what are adjuvants? These are the substances that can enhance and improve the immune response to an antigen when combined or mixed with the antigen. Directly immunizing with most antigens will lead to poor immune response and rapid removal of the antigen from the body. So adjuvants are mostly used in vaccines to boost the immune response in case of weak antigens or when the antigens are available in small amounts, thus limiting the dose requirement of the antigens. Adjuvants create a depot effect for the antigen, slowing down its release and thus prolonging its persistence on the site of injection. They also help to enhance the secretion of cytokines and chemokines, thus maximizing the immune response before the antigen is cleared by the body. Common examples of adjuvants are aluminum hydroxide, aluminum potassium sulfate, Freund's complete and incomplete adjuvants. Freund's incomplete adjuvant contains aqueous solution of the antigen and mineral oil and an emulsifying agent such as mannite monooleate, which disperses the oil into small droplets surrounding the antigen, thereby slowing down the release of the antigen. Freund's complete adjuvant contains heat-killed mycobacteria in a water in oil emulsion. The muramyl dipeptide component of the mycobacterial cell wall activates macrophages, thus making Freund's complete adjuvant much more potent than the Freund's incomplete adjuvant. Now here is a practice test to evaluate your understanding of the topic. 10 multiple choice questions are given and you have to choose the correct option for each. Question number 1. An incomplete antigen is also called as hapten, is immunogenic upon binding to a carrier protein, cannot induce antibody production by itself, or all of the above. Question number 2. Antigens can be proteins or carbohydrates or nucleic acids or all of these. Moving on to the next question. In humans, blood group antigens A and B are example of heteroantigens, alloantigens, neoantigens, or heterophile antigens. Question number 4. Majority of antigens are proteins or polysaccharides or lipids or nucleic acids. Next question. Any molecules that induce or elicit an immune response are called antigens, antibodies, epitopes, or immunogens. Question number 6. Which of the following statements is incorrect about superantigens? They are viral or bacterial proteins, endogenous by nature, have unique binding ability, or activate a large number of T cells. Question number 7. Which of the following statements is true regarding adjuvants? Adjuvants decrease the efficacy of vaccines. More antigens are needed when an adjuvant is added. More antibodies are produced when an adjuvant is added. Or more doses are needed when an adjuvant is added. Next question. Immunogenicity of a substance increases with phylogenetic distance between organisms, molecular weight, chemical complexity, or all of the above. Question number 9. Which of the following is an example of endogenous antigens? Fungi, protozoa, viral proteins produced during viral replication, or bacteria? Now the last question. Which of the following is not true for T-dependent antigens? They are proteins, induce immunological memory, are recognized and presented by APCs, or directly activate B cells to produce antibodies without T cells' help. Here is the answer key for all the above questions. You can match your responses and check your score. Here are some of the references that I have used to make this presentation. Thank you.